For those of you joining us on this Google Hangout, good afternoon. I'm Kevin Robert Frost. I'm the Chief Executive Officer at amfAR, the Foundation for AIDS Research. And I'm joined by Dr. Rowena Johnston, amfAR's Vice President and Director of Research. We're here today to talk to you a little bit about the case that has been in the news. I'm sure most of you have seen it, otherwise you wouldn't be here, about a child that was cured of HIV infection. We're going to chat, Rowena and I are going to chat for about 15 minutes or so, just to bring you up to date on what we know about the case, and then we're going to open this up for questions. So if you do have a question, you can either ask that on Facebook, or you can ask it via Twitter, at amfAR, via Twitter, or just go to the Facebook page for amfAR and put your question in there. So I think we'll go ahead and get started, if that's okay. And why don't we start by having you go back to the beginning and describe the case for everybody.
Yeah, you know, this is a really interesting case, very thought-provoking. This really all started when a mother presented at the hospital. She was in labor, and she was presented at a hospital in rural Mississippi, and she was tested during labor for HIV because it was clear that she had not been in prenatal care at all. So the doctors administered a rapid HIV test and determined that she was, in fact, HIV positive. And so from that, the pediatricians in care of the case knew that the baby was at fairly high risk for acquiring HIV. And so after the baby was born, they decided to administer a treatment dose of antiretroviral therapy to this infant. This is actually probably the key that makes this different from a lot of other cases of HIV around the world resulting from mother-to-child transmission. And that is that there was a treatment dose of antiretroviral therapy. Normally we would try to give prophylaxis or prevention, but it was a treatment dose. And, you know, that was really a very interesting decision that she made. Around that same time, they were conducting tests to determine whether or not this infant really was HIV positive. And by that time, the infant was transferred to the University of Mississippi Medical School, where they were able to do some tests that really did determine that the child was HIV infected. By that time, she had already started a treatment dose of antiretroviral therapy. And, you know, really for the next 18 months or so, she was in medical care, the way a lot of infants in the United States or around the world might be in medical care for HIV. But at 18 months, the mother and child stopped coming back to medical care. And they actually reemerged about 5 months after that. And that was when this story really became very interesting.
Why? What happened?
So the child came back, and of course the first thing that the pediatrician wanted to do was to determine how much virus is now present in the child because she hasn't been receiving treatment for all this time. She administered, the pediatrician, Dr. Hannah Gay, who works in Mississippi, administered a viral load test, and she found that there was no virus there. And the first thought that crossed her mind was, well, I must have made a mistake here, so I'll do this test again. And she re-administered the viral load test, the kind of test that anyone in clinical care would get, and yet again, the test came back negative. And this was the point at which she knew that something very unusual and interesting was happening, and she decided to call on her colleague, Dr. Catherine Luzuriaga, who works in Massachusetts at University of Massachusetts Medical School, and told her a little bit about this case because she actually wanted to get advice. How should I treat this infant now? I mean, what do I do now? I haven't seen this before. And Dr. Luzuriaga and Dr. Deborah Prasad, her colleague, were then both called in to consult on this case. And as luck and fate would have it, the two of them had received an amfAR grant to look at exactly cases like this. And so they were able to employ all of the best scientific minds in the world, really, that specialize in looking for exactly this kind of thing. Does this child have HIV? And so blood samples were sent to various laboratories, 6 laboratories around the country. Everybody used their own test to see whether or not they could find any HIV in this child. And nobody was able to find any convincing evidence that this infant was still HIV positive.
So let's back up for a second, because I want to unpack it now a little bit and get in a little bit deeper into the case. When the mother arrived, she was already in labor?
She was already in labor. She arrived really at the point of labor, and her labor, they had time to administer an HIV test, but the labor, in fact, was so rapid there was not even enough time for them to administer antiretroviral therapy to the mother, which would normally be done, but the labor was so rapid that they just didn't even have time to do that.
So the mother didn't know she was HIV positive.
That's right.
She found that out at the time they did the test
at
when
that
she
moment.
came in. I
Right.
see. So the child was born, and the mother didn't use any antiretrovirals in that process at all. That
That's
would be how
correct.
you would normally prevent mother-to-child transmission.
Yeah, in the United States, women would normally be in prenatal care, and as part of regular standard prenatal care, women are administered an HIV test because we know that women who are HIV positive can take antiretroviral therapy, and in almost all cases, really only the exception of 1 or 2% of cases, in almost all cases, you can prevent the transmission of HIV from the mother to the infant if the mother takes the antiretroviral therapy during pregnancy. That didn't happen in this case because this mother had never been in medical care until that moment that she came into the hospital during labor.
So is that part of the reason why doctors felt strongly at the time that this child was at high risk for infection?
Yeah, that absolutely was a huge contributing factor. They knew that the mother had not taken any prophylaxis. There was nothing that had been done to prevent HIV transmission. And so rather than playing it safe and administering prophylactic or preventive types of antiretroviral therapy in the infant, they decided that really the risk benefit was such that let's go ahead and administer a treatment dose because that's really probably in the best interest of the infant to really just start her off on this treatment dose, and let's wait and see how the tests come out after that.
So how soon after the child was born did they do the first HIV test?
They drew the first blood sample at 30 hours to determine whether or not the infant was infected, and then they drew a separate sample at 31 hours. And each of these samples of blood was tested to determine, is the child infected? And this is standard practice also in the United States. This is exactly how you determine, is a baby infected? Because one of the complicating factors for even diagnosing HIV infection in infants is that every infant is born with its mother's antibodies. So you can't administer a standard HIV test to work out whether an infant is infected. What you have to do is take blood samples and do somewhat more complicated laboratory-style tests, looking both for what we associate with RNA and DNA. Those tests were done in this infant. Both of them came back positive. The infant met all of the standard definitions for infection by 31 hours of age.
So the tests that were administered were looking for viral DNA and viral RNA in the infant.
That's correct.
Not antibodies.
That's right.
So is that what gives you the confidence that the child was in fact infected, those two tests, or is there more?
Well, I mean, certainly there is more, but I want to emphasize that, that is the standard definition. That is the universally applied definition for infant infection. You have two separate blood samples, and you have the DNA and the RNA, both of direct evidence of the presence of the virus. That alone is sufficient to tell us that this child was infected. But in fact, because the child was put on antiretroviral therapy, further standard practice is that you monitor how well is this infant doing on the therapy. And so at repeated intervals during the first 3 weeks or so of life, the infant had more blood drawn, and those tests were done to see, are the virus levels going down? Because that's, of course, what you hope for when somebody's taking antiretroviral therapy.
To show that the virus is responding to the treatment.
Exactly. And those repeated tests were done. Those during the first 4 or so of those tests during the first 3 weeks of life also indicated that the child had virus in her, and those levels were going down, as you would expect if somebody's responding well to antiretroviral therapy. And so we really do have a whole series of tests that tell us the child really was infected, and she did respond well to antiretroviral therapy, and by 3 or 4 weeks of age, she had reached what we call clinically undetectable, which means that the standard test that a doctor would administer in clinical practice no longer is able to detect the virus. And we assume, and what is almost always true for adults or infants, is that the virus is still there, just a little bit below the range of what the test can see.
If this were just another case of mother-to-child transmission, or prevention of mother-to-child transmission, would you have expected to see the virus positive on those tests that you talked about?
Well, no, we wouldn't have. I think, the fact that we have those two tests, one at 30 hours and one at 31 hours, both of which confirm that the infant was infected, and then I think really tellingly, the fact that over time, in response to the therapy, we have repeated measures where the virus continues to decrease in exactly the way that we would predict to have happened, that's really what gives us the confidence to say the child was infected and responded exactly, at least initially, the way we would anticipate to antiretroviral therapy.
So how does a newborn, not how, but when is a newborn infected? Are they infected while they're still inside the mother's womb, or are they infected during the birthing process itself?
You know, that's an interesting question because there are actually 3 stages at which we think of mother-to-child transmission. One is what we call in utero, and that is sometime during pregnancy, probably during the last trimester, but it's harder to pin down any more precisely than that. An infant can become infected. They might also become infected actually during the birthing process itself, and we call that peripartum transmission. And the infant, of course, can also be infected by breastfeeding after birth. We do know that this child was never breastfed. That much we know. But how do you distinguish between a child that became infected during the pregnancy versus during the birthing process itself? And this child actually met the definition for having been infected during pregnancy, because you would not expect a baby to show up on these 2 DNA and RNA tests. You would not expect the child to show up at positive so early after birth. In the first 48 hours after birth, the positive test really only happens if the infection happened during pregnancy and not during the birthing process itself. So we don't know for sure when this child did become infected. I mean, I guess we don't really know for sure that it didn't happen during the birthing process, but the chances are that it actually happened sometime during the 3rd trimester.
So what's the best theory here about what happened? How was this child cured?
Yeah, I mean, that's the million-dollar question, right? Because, you know, we have to work out what made this child different from all the other infants that we know have been born in the United States as well as around the world with HIV infection. In terms of the treatment, we know that the child received a very early dose of treatment-level antiretroviral therapy. But why did that work? That's really what we want to know. What we think happened is that having the antiretroviral therapy, those drugs that counteract the ability of HIV to reproduce, having those on board so early actually stopped the virus from being able to establish what we call reservoirs. And the easy way to think of reservoirs, if you imagine a person who's been infected for a number of years, say, they can most often get their virus levels down very, very low if they take antiretroviral therapy. And in fact, we think that the virus levels are so low that the virus actually stops turning over for the large part, is not able to replicate anymore. And yet, as soon as that person stops taking antiretroviral therapy, the virus levels bounce back. What we know then is that there are reservoirs of virus inside the body, inside specific cells, perhaps inside specific areas of the body itself, that harbor the HIV. And the HIV inside those cells is impervious to the antiretroviral therapy. And we know that these reservoirs are established very soon after infection. I think actually we've learned something very interesting about the establishment of reservoirs in this infant, because she received her antiretroviral therapy starting around 31 hours of age. And I think what this tells us is, in all likelihood, the antiretroviral therapy came in before the virus had an opportunity to establish these reservoirs. So we've learned something about the timing of when these reservoirs get laid down. And I think that's actually going to be very informative on looking at how might we cure other ways in which HIV is transmitted.
So you said reservoirs can be formed either in anatomical regions.
That's right.
Examples being...
In the brain, for example.
The brain.
As a key one.
But also in cell lines, for
That's
example.
right.
They can form in different types of cells. Is there a particular cell line where it's believed reservoirs form?
Well, we know that HIV has a preference for infecting what we call CD4 positive T cells. This is a certain type of immune cell that really is kind of what we think of as the conductor of the orchestra, the one that really kind of tells the rest of the immune system how to respond to different infections that enter our body. And we know that HIV has a particular preference for those cells. But then you can actually even divide those cells into further little subcategories. There are central memory T cells and transitional memory T cells and effector memory T cells. And what we believe is that in most cases, the reservoirs are largely found in central memory T cells. Most studies, people who acquire HIV infection as an adult, they've been infected for a few years, you do these very detailed studies looking at very, very low levels of virus that persist, it really does look like they persist largely, not entirely, but largely in central memory T cells. So what's interesting is that a baby is born without these central memory T cells. So what we don't know is where was this virus that she was born with hanging out.
You're saying children are born without these central memory T cells, therefore without the reservoirs themselves.
They're certainly born without the reservoirs. What's interesting, we're treading exactly on that territory that a lot of more research is going to have to work out for us. Which cells was the HIV replicating in this infant? Why did it not have a chance to establish reservoirs? How different is the immune system of an infant to an adult? And what was it that made the difference that this was possible in this infant? Let's look at how that knowledge can be transferred into adults.
So what does this mean for the immediate future now? Everybody knows the Berlin case. Everybody knows Timothy Brown. Now we have a child case, which looks incredibly promising. What are the implications of this case? What comes next?
Yeah, I think we have to keep in mind that this is one case. So what we need to understand better is, was there something in particular about this infant that allowed this to happen in this infant, or is this more widely generalizable? And certainly researchers are going to be very interested in conducting clinical studies to look at whether treatment in infants this early after birth might actually have the same effect. That would be the first step. And I think, after that, we are going to be very interested in understanding these key differences in the immune system of an infant versus an adult, understanding exactly what role do the reservoirs play, when do they get established, how can we get rid of them, because that's how we're going to cure people.
So are there any immediate implications? UNAIDS says that there are more than 330,000 children born every year with HIV around the world. Are there immediate implications for those 330,000?
I think one of the interesting things about this case is that a lot of women in the developing world, for example, are also not in prenatal care, the way this mother in the United States was also not in prenatal care. So although we know how to prevent mother-to-child transmission if the woman is engaged in prenatal care, there are still hundreds of thousands of infants born around the world because these women don't have access to antiretroviral therapy, for example, during pregnancy, and maybe only present during the process of labor itself. So what this means is, depending on whether these results pan out, and this really is generalizable, we may have something that could really be immediately implemented worldwide, where a woman who really only does present at labor rather than during pregnancy gives birth, and we've determined that she's HIV positive, and we know that the infant is at very high risk for HIV. These are antiretroviral drugs that are available around the world. Maybe we could administer those to these infants within this short period of time after birth, and we then are able to prevent a child from having to live an entire lifespan with HIV, and also are able to make it such that that person doesn't have to take antiretroviral therapy for their whole lives. Clearly the social and psychological and economic implications of this really have the potential to be huge.
So one last question, just briefly. Could you clarify for me one more time, and clarify for the people that are watching, what amfAR's role was? Because obviously early on, when the child was discovered and put on therapeutic dosing and everything, that was all before amfAR got involved.
Yeah, that's right.
Just clarify really quickly what amfAR's role was.
Yeah, Dr. Hannah Gay, the pediatrician, got in touch with Catherine Zuriaga in Massachusetts, who in turn got in touch with Debbie Prasad. The two of them are leading a consortium of researchers funded by amfAR and set up to look at cases exactly like this, both to look at new infants infected with HIV to see what we can learn about how to cure infant infection, but they had also set up this collaboratory of researchers all around the country, all of these people specializing in their own tests to determine whether or not a person is cured of HIV. When this infant came on the scene, that collaboratory was already in place. And so immediately, this group of researchers was able to jump on these blood samples to take a look, determine, can we, by the absolute most sophisticated means we have available to us anywhere in the world today, can we find any HIV in this infant? And as a group, they were able to, within a month or two of this case emerging, all come to the same conclusion: this infant no longer has HIV.
So it's fair to say that amfAR's funded research is what really confirmed the case and provided that
absolutely
structure.
what confirmed the case. These are tests that are not conducted in the clinic. This is really something that has to be done in a sophisticated lab setting. We had funded this setup so that that sophisticated laboratory resource was available and was able to confirm that this child was cured.
Great. Let's take some questions because we do have a couple of questions coming in on Facebook and Twitter. Again, if you want to ask a question, you can tweet at amfAR or you can go to amfAR's Facebook page and ask the question there. So the first question is coming in from Twitter, and it says, how does the news of the first child cured of HIV impact the approach to one day curing adults?
I think we're going to have the potential to learn a lot about the establishment of reservoirs and what it means to eradicate reservoirs, for example. Clearly, I think the leading hypothesis in this case is that what was important is that we did not allow the establishment of reservoirs. And so we're going to be able to learn how do we translate that into the adult setting. We do know that there are significant differences between the immune system of an infant versus an adult, and so the findings might not be immediately transferable in the same way, but the fact that this cure has happened has obviously lifted people's spirits. It was done in a way that is really, if it is generalizable, it's going to be a lot less complicated than the Berlin patient case. And so I think it's given people a lot of energy, and it's given people new clues as to which kind of cells do we need to look at, which kind of immune responses do we need to look at in order to cure people.
Terrific. Question from Facebook. I'm interested in the research and development of latency reversing drugs. What's the latest, and what impact does the functional cure of this infant have on this research?
Yeah, I mean, as we were just discussing, this infant was cured seemingly by antiretroviral therapy, and this is something that's already available, and I think that's really one of the exciting elements of this case. Now, we don't know to what extent this information can be applied in adults, and we do know that there is research ongoing in all the different ways in which we might be able to cure adults, including gene therapy, including immune interventions like therapeutic vaccines, but also including these latency reversing drugs. That research is ongoing. Certainly amfAR is funding some of that research. There's a new clinical trial that's going to be starting any day now, literally. And other centers are also conducting that research. We're going to have to wait and see what those results tell us.
amfAR's funded a little bit of research into some of these latency stimulating drugs, if you will. Is there anything new on that front?
Dr. Lewin presented some results at the recent conference in Atlanta. We do know that, like I mentioned, amfAR is funding a new clinical trial of disulfiram, one of these drugs. And disulfiram is a very interesting drug. It's a drug that was discovered to have an anti-latency effect by one of our grantees, Dr. Robert Silicano, who works at Hopkins University. And nobody, of course, would have anticipated that an anti-alcohol drug might play a role in HIV cure. But a first small clinical study was done in which there were some hints that this really may have some promise in the context of curing people. And so what we needed to do is to work out, how can we get enough people in a study where we can really work out what's going on. One of the great things about this trial is that because it's a drug that's been on the market for 50 years, we already know it's safe for people to take.
Yeah.
So we can just get straight into those trials and let's see what happens.
Terrific. Another question from Facebook. How readily available will it be in the near future, and what can we expect to see in the form of statewide funding and insurance coverage for this cure? Well, obviously the Facebook writers are getting a little ahead of themselves, aren't they? I mean, this isn't a cure that's deliverable at this point yet. It still needs to be confirmed.
It does need to be confirmed. I think it would be deliverable if it's confirmed. But we need to make sure that we're not getting ahead of ourselves. This is one case. It's a very interesting case. It's a very exciting and very promising and very thought-provoking case.
But it has to be replicated.
It absolutely does.
Right.
And we know that the researchers are already making plans on how that might best be done. Clearly, it's going to be a high priority for a lot of people to make sure that we can replicate it. And once that is done, I really do feel optimistic that this can be put out there.
Good. From email, did the doctor who decided to treat the baby with the 3-drug regimen make that decision as a result of amfAR research? Well, I think we've already said no to that because that happened very early on.
It did happen very early on. She made a very interesting decision to do that, and it's possibly what made the difference in terms of actually bringing about what happened in this child. But as we've discussed, what amfAR's role here was to be able to fund the research that confirmed that this is a cure, because it's very sophisticated testing that needs to be done,
right.
And that's what amfAR's funding was able to do.
But it's fair to say, I think, that the child itself was cured largely by accident.
It was a case of serendipity, and
Yeah.
a lot of science happens that way. Our goal now is to learn from that. I mean, we don't want to let this opportunity pass us by. Now that we've been delivered of this serendipitous and happy accident, we need to work out what happened, and let's see what we can do in the future.
Can the first child cured news confirm that we are close to a formula for the cure?
You know, there are different ways of looking at this question. This is an infant cure that happened, I think, because of a set of circumstances that need to be confirmed. Can this be done in infants on a broader scale? There may be some difficulties in terms of transferring this to adults, in as much as adults often don't know, as soon as this infant did, so to speak,
right.
that they're infected. You know, we still have a lot of challenges getting people to be HIV tested at all. And even if you had a standard HIV test, it wouldn't show you that you're infected within this window that we're talking about in order to start the antiretroviral therapy and then to have an effect. So, you know, there are a lot of things that would need to be changed, but a lot of things that can be learned from this case.
Great. From Facebook, this is a really good question because this has come up in other conversations. It seems there have been previous cases published in medical literature suggesting other infants have been cured of HIV in the past. What's the difference between those cases and this one?
Yeah, and in fact, starting probably around the mid-1990s, there had been sporadic cases reported in the literature where it did appear that babies may have been cured, or even, you know, really just kind of spontaneously cleared HIV infection even without antiretroviral therapy. The difference here, and this is actually the difference that the amfAR research made, is this case is incredibly well documented. There have been researchers who've gone back to look at those earlier cases, and really in every case they've been able to determine that the samples were contaminated or damaged to the extent that you really couldn't do the testing that's needed to confirm that the infant was really infected in the first place or cured later. Also, we're talking about a time where the sophisticated tests were not actually available yet.
Right.
And so this really is actually the first well-documented case of an infant cure.
Dr. Prasad, who was the lead author in Atlanta, also spoke to this issue, and she talked particularly about a paper by Bryson et al., which had published a number of these cases, and she said that when the forensic medicine was done on all of those cases, it showed that 99% of them could be proven to be either laboratory contamination or laboratory error in the testing. The other thing I think that's very telling about it is that since that time, there's never been another recorded, since the appearance of PCR, there's never been another recorded case of spontaneous cure. So I think there's a fair amount of evidence to suggest most of those were laboratory error.
I think that's absolutely right. Just didn't have the mechanisms in place to really determine with the same certainty that we have today.
Fair enough. I think we have time for just one more question. When there is a cure, will amfAR take the role of making sure it's available globally? Well, sure, I'll take that. It's a fair question, I think, although, again, I think we might be getting ahead of ourselves a little bit to answer that. There's no question that whatever form the cure takes, it will require an enormous infrastructure to deliver it. We know that now just from the work that's going on around polio, the effort that it took around smallpox to vaccinate the world. With more than 33 million people living with HIV, delivering a cure to 33 million or more, whenever it arrives, will be an enormous undertaking. But amfAR's job is about research. It's always been about research, and it's understanding that our job is the quest for answers. That's what we do. So it is my sincere hope that when that day finally does come and we finally do have the cure for this epidemic, that we'll close our doors and go home. because I think the job of delivering it will then become the job of governments and other kinds of aid organizations who specialize in that kind of work. And that's a very different kind of work than what we do here. We're all about biomedical research and the policy and human rights that surround that. So I think when we have a cure for AIDS, our job will be done. Thank you all very much. We appreciate you coming to see us today. Please send us your comments. We'd love to hear from you either on Facebook or Twitter or email. If they're negative comments, email Rowena. If they're positive comments, feel free to email me. I love those kinds. Thanks very much for this. We appreciate it.
Thank you.
Thanks.